Hello, my name is Sola Gbenro, and this is CaoLab2 protein function prediction using hidden Markov models. So I'd like to start with the problem, which is that the disparity between, or the gap between, known protein sequences and knowledge of their function is increasing rapidly. Two of the biggest reasons why this is that it's relatively inexpensive and fairly quick to determine a protein sequence nowadays. But it is still rather difficult and fairly costly to accurately determine a protein's function. So the question is, given a protein sequence, can we accurately predict the function by associating it with a gene ontology term? Our tool attempts to solve this problem and shrink that gap between the known protein sequences and their function using a hidden Markov model approach. Our model uses Go term language created by performing a depth-first search on each gene ontology tree to index the term and convert them into a character string called a Go word. After matching the UniProt database with over 536,000 sequences to said Go words, the result was 27,451 unique words in this Go language. This large discrepancy between the Go words with the most amount of associated sequences and the Go words with the least amount of associated sequences created an imbalance within the representation of our words within our dataset. As a means to correct this, we employed data augmentation to artificially inflate the number of protein sequences belonging to Go words that have less than 100 sequences associated with them. For these words, we trained a separate hidden Markov model, or an HMM, on their available data and used this to generate new sequences up to the minimum of 100. We introduced some variation within these artificial sequences by allowing for lengths that were within plus or minus 10% of the average sequence length for that specific Go word. For Go words with sequences between 100 and 1,000, all the available data was taken. And for Go words with greater than 1,000 associated sequences, a random sample of 1,000 was pulled. Now, HMMs can be very computationally expensive, and even with the computing power available to us through Google Cloud Compute, this method utilized 400 CPU cores for 3 weeks to train the 27,000 models that were required for a comprehensive Go term representation of our Go term list. In function prediction, our first step is to acquire a FASTA sequence. When we take the sequence, we predict on all the Go terms, ranking them by highest probability. We only accept the models that produce a score within 80% of the top scoring model, or a total of 200 models, whichever is less. This list is then taken as the function's prediction with the top score assigned a value of 1.0 and the remaining values adjusted respectively. We tested 253 unique sequences selected from the UniProt database. We performed tests using the top-end metric, which produces statistics for predictions ranking 1 to 20 for each target. We analyzed the maximum and the average similarity between the top-end set of predictions as compared to the true set of the Go terms for each target. Results shown in the tables here, they compare the sequence similarity-based methods in blue, the network-based methods in green, and sequence-based methods in red, as well as the information-based method in black. From the maximum similarity graphs here, we see that the maximum similarity for the biological process subcategory for HM meta outranks the other sequence-based methods and even manages to exceed the network-based method. Now this slide, this is the maximum and the average similarity between the predicted set and the ground truths for the same sequences, but taken as a whole. HM meta produces results similar to other prominent sequence-based methods. With some tuning, we believe that we could continue to improve this method and correct for some of its pitfalls. Future research and some possible adjustments that we'd like to make include around the data augmentation. Several Go words only had one associated sequence with them, which means that there was very little data for the sequence predicting HMM to train on. This also means that the vast majority of the associated sequences were artificial. A possible solution to this could be to do a multiple sequence alignment beforehand to find close relatives of the available sequence, and then include those relatives in the training data. Additionally, we were limited to only 5 hidden states for all of our HMMs due mainly in part to the resource costs and the training times. A technique to supplement our results could be using an ensemble method and combining results from several of the models to increase our accuracy. Lastly, by benchmarking against additional methods, we would gain a better understanding of how our method compares within the industry. I'd like to take this time to acknowledge Kyle Hippie for his perseverance, his in-depth knowledge, and his willingness to provide all of the support in the world. And to Dr. Cao for introducing us to the world of bioinformatics and his guidance and mentorship through this journey. Thank you.